This will be the first of two talks talking about applications potentially for QSTORM. And we're, I don't think anyone has said this, we're probably about halfway into this planned 3-year project. So you're definitely seeing work in progress in these talks. We haven't yet gotten to the point where we want to be, but we're excited about the potentials for this collaboration and for QSTORM imaging. So striated muscle has just a beautiful structure, beautiful repeating units of myofilament proteins. The traditional way of looking at muscle proteins is with transmission electron microscopy, and you get these beautiful bands of light and dark muscle proteins. This is the thick filament in the dark middle band here. This is the so-called Z line or Z disc, which differentiates the repeating units, the sarcomeres of muscle. In this region is where the actin is, this is where the myosin is, and a whole terminology has arisen based on these light and dark bands that describe these, this banding pattern. So the A band is the region that contains the myosin, and it has these black myosin heads. A region that has no myosin heads, more myosin heads. These are C proteins that are also visible in the TEMs, region of actin, which is the I band, and then these Z discs. So there's, as I said, a region, an M region or bare zone where you don't see cross-bridge heads. And then there's another very important protein. There are many other proteins, but one of them is titin in these sarcomeres. And these titin molecules actually span the whole length. And we're just seeing the part of it where we can see it in the I band here. And it's thought to be the scaffolding on which these muscle proteins assemble to make the repeating units. To just put this in the scale of a whole muscle, so if we have a whole muscle body that is organized into fascicles, which contain the muscle cells, the muscle fibers, and those contain myofibrils, and those contain these muscle proteins. In terms of scale, the length of a sarcomere is typically about 2.1 microns, but then the actual diameters of these muscle proteins is much, much less. So on the order of 10 nanometers for the thick filaments, for these red thick filaments in myosin, but 5 nanometers for the actin and 3 to 5 for the titin. So we're really talking about an interesting multi-scale problem. We're interested in very small diameter proteins, but that are very long. And so how do we image something like that at those multiple scales that are interesting? So we can see quite a lot of this, at least banding structure, not just in TEM, but also with immunohistochemistry and confocal microscopy. So here we've done a double label for the myosin heads and titin. And so we can see this red region is where myosin heads are. We can just barely see a bare zone here where there aren't myosin heads. And then we see the titin in that I band, and then we see the Z disc in the middle of that I band. So just to label some of those things, we can see the Z line or Z band, we can see the M line, and we can double label and see what's happening with multiple of these proteins and how the lengths of these regions, for example, might change during contraction. So the standard model for muscle contraction is the sliding filament model, which was developed over half a century ago. It's a really powerful model quantitatively in terms of its predictions of muscle mechanics. We can predict almost everything about the active behavior of muscle from our understanding of actin and myosin sliding past each other, the myosin heads, the cross bridges pulling on the actin. This was developed from TEM by the fundamental observation that the thick filament doesn't change length. The thick filament stays the same length if you're looking at fixed muscle that's fixed in the contracted state and fixed in the non-contracted state, but these Z lines come in when the muscle contracts. The filaments slide, the Z lines come in, and that's the cross-bridge heads pulling on the actin. And so this has been a pretty powerful model for a long time, but there have been aspects of muscle mechanics that aren't very well explained by this model. The curves don't quite fit. There are aspects particularly of when we stretch active muscle, and there are aspects of time history dependence that have been hard to explain with the simple sliding filament model. And what we're finding is that there's maybe a lot more going on in there, and particularly with titin. So here we see the titin is within these thick filaments, but then it branches out in the I band, and there are particularly these really fascinating N2A sections that it turns out have a calcium activation. So we always think of calcium activation as producing the cross-bridge head motions and the sliding, but it turns out calcium does a whole lot of other things in muscle, and one of them is to cause this N2A region to bind to the actin. So this N2A in the presence of calcium, there's really excellent evidence that this N2A binds to the actin, and that changes the length of this PEVK section, which acts as a spring. And so the muscle actually becomes passively stiffer in the presence of calcium, even if you disable the cross-bridge heads, because you're essentially shortening. This is a long spring. You connect that. You've got a shorter spring. It becomes stiffer. But then there's this new theory from Kisa Nishikawa that the titin might actually be winding up on the actin. And this is at this point, it's purely theoretical, but she can explain certain non-constant stiffness properties of muscle based on the idea that this PEVK region, this spring region here, would actually wind up and change its stiffness even more, because it would make it effectively an even shorter spring. And so if muscle has been active and this is wound up, that would add some historical knowledge essentially to the sarcomere about what's happened to it before, which would affect what happens later. But at this point, this is purely a theory. It's an exciting theory that needs to be tested. It suggests that something interesting might be going on in the Z bands where these actin molecules are anchored. And for quite a while, we've known that there are interesting dynamic things that go on in the Z bands, that when muscle activates, it goes from in TEM from a square pattern to a basket weave pattern. So here we have actin filaments, and we have alpha-actinin filaments that are anchoring those in the Z disc, and that pattern changes when muscle is activated. We don't exactly know what that means, but it's very intriguing when you think about it potentially in the context of that winding filament model. Another thing that people have been focusing on recently, which is really interesting, is the idea of what happens at the molecular level to bulging when muscles contract. We've long known that if you contract your muscles, they bulge out. But if you think about that at an actin and myosin level, it means that as those filaments are sliding past each other, they're also moving slightly further apart from each other, and that's affecting what's happening in the Z discs as well. And so just the recognition that if we look at this now in cross-section instead of the usual sarcomere longwise, if we look at the cross-section at the myofilament lattice, that this myofilament lattice is going to be changing as the muscle is contracting. And so there's some new modeling looking at cross-bridge head dynamics in the context of muscle bulging at the molecular level that is actually explaining fundamental things like length tension of muscle better than we used to be able to. So there are a lot of things right now going on in the theory of muscle contraction that suggests that it's more than just sliding filaments. The sliding filament model isn't wrong, it's just there are other things that we could add in that would explain a wider range of muscle properties better than just actin and myosin sliding filaments. So as I said, with standard confocal microscopy, immunohistochemistry, we can see quite a lot of this structure. But in order to really get down to, for example, labeling just that N2A section and seeing how it changes relative to Z disc positions and thick filaments, we really need more resolution than we can get from standard confocal microscopy. And this is where super resolution imaging and potentially using the quantum dots for labeling comes in, and the collaboration of our QSTORM group. And one of the first concerns I had when I thought about using quantum dots for muscling label is, can we get those quantum dots, and particularly things like those larger micelles, into that myofilament lattice, right? Is there space to get them in? Do they intercalate themselves between the myofilaments, or do they just stay outside and will be, create a real problem for labeling? So can we get the Q dots into the myofilament lattice? And so we started testing this in zebrafish because we were interested in using the quantum dots for in vivo labeling. If we want to be able to see muscle contraction, we need something that will contract. So we were thinking to do this in zebrafish larvae. And this is just an example of Texas red micro-injected into a single muscle cell of a 5-day post-fertilization zebrafish. This is an untargeted label at this time, but the goal eventually would be to label actin, myosin, titin, eventually with quantum dots. So far, we've done these microinjections in living zebrafish larvae. We've microinjected untargeted fluorescent dye into single muscle cells. We've microinjected untargeted quantum dots into single muscle cells. And when we did this, we find that they tended to spread out a lot. And this could well be just bad microinjection technique on our part. It could be the osmolarity of the injected solution, but it still allows us to look and see whether these quantum dots are intercalating into the myofilament lattice. So here's just how this looked in the confocal microscope. The dyes tended to stay right in the single muscle cell. The quantum dots, which were the micelle-coated quantum dots that Juan was just talking about, tended to spread out more. But the wonderful thing about quantum dots is they're also electron dense. So we can look at them in TEM to see where did they go. And what we found was that they did appear, even those relatively large micelles did intercalate themselves, did diffuse down in between. So here we have a Z line, here we have a Z line, and here's one sarcomere, and you can see these quantum dot micelles have intercalated themselves down into between the myofilaments. So it seems like maybe we don't need to be quite as concerned as I thought we needed to be about the size of these things, although of course I'm still concerned because when we talk, if we're sticking all these things in there and then we have these things contract, it could be interfering with contraction. We have yet to see how that's going to work. But there are also real challenges with working with the zebrafish system in vivo for this, because at this point these were untargeted. We weren't doing any particular labeling. And in order to do labeling, we need to label and then somehow wash away the unbound label in order to do very specific labeling and with quantum dots. And so we're moving to a rabbit psoas isolated muscle fiber prep. And these rabbit psoas fibers are just amazing. They take them out of the rabbits. They put them in glycerin. You store them at -20. We actually get them from Carolina Biological. And the glycerin permeabilizes it so that we can get the antibodies in. We can wash away the unbound antibodies. But these things still contract for months. So we put them in a solution with ATP and calcium, and these muscle fibers will contract. And so we're able to actually, so they're unfixed muscle fibers, and so we're able to make them contract. And here's a double label again with myosin and titin of a contracted rabbit psoas fiber. This is just a Z projection, maximum intensity projection through a Z stack of these things. And so we are able to do the labeling so far. This is with Alexa dyes, but we don't see any reason we couldn't use a quantum dot secondary here and be able to label and wash away the unbound label and still look at muscle contraction specifically bound to the proteins of interest with quantum dots. Of course, if we were, we could do some sort of expression for GFP, that would be another way to go. But if we're going to be trying to use quantum dots, we need to get them in from the outside and wash away the unbound. So next steps, we're going to be doing quite a bit of STORM imaging with just the Alexa dyes while we're figuring out the labeling with the quantum dots and how the quantum dots might potentially be affecting the contraction. We're going to go ahead and do some of this work with STORM imaging with Alexa dyes. So we'll be able to label the myosin, the actin, titin, just even that specifically that N2A region or the PEVK region, alpha-actinin to look at some of the dynamics in the Z disks. And with super resolution microscopy, we'll be able to look at the dynamics there. And we're working with Kisa Nishikawa, the person who's created that new crazy winding filament. We're working with her to try to come up with some definitive imaging tests for if that's actually winding, what would we expect to see if we could get down to, say, 50 nanometer resolution. And so, as Peter showed, so far we are getting the Alexa dye STORM to work. So that's just a confocal image, and then a more magnified. That's a 1 micron scale bar, and that's a 1 micron scale bar. More magnified. We still have work to do to get the storm images to show us the biology we need to see, but it is looking very, very promising. Next thing will be to develop a Q-dot secondary in the rabbit psoas model so that we can actually Q-dot label, and then finally to develop the dynamic muscle contraction methods so that we can image the very same region of a fiber before and after contraction to see what the changes in these muscle protein relationships are. So I'd like to acknowledge Callie Harper and Julia Olszewski, the students who've been working on this. Julia is an undergraduate at Brown, and Callie is a doctoral student. I'd really like to acknowledge the Innovations in Biological Imaging and Visualization Ideas Lab that brought our group together. This has been taken certainly my research in directions that I didn't really expect to go right now, and it turns out to be just a great time to be getting into imaging muscle at the molecular level. There are all these interesting models and realizations out there that if we could actually image these things, it would be great to test those. And the whole QSTORM collaboration has been great, so thank you.